By the third day after fertilization, the embryo has developed into a solid ball of approximately 16 to 32 cells, known as the morula.
As it continues traveling through the fallopian tube toward the uterus, fluid gradually enters the morula, creating a fluid-filled cavity.
This transformation produces the blastocyst, a highly organized structure that represents a critical milestone in embryonic development. The blastocyst consists of two major components. The outer layer of cells, called the tropho, later contribute to the formation of the placenta and other extraembryonic tissues that support pregnancy. Around the 5th day after fertilization, the blastocyst breaks free the surrounding zona pellucida in a process called hatching. This step is essential because the embryo cannot attach to the uterus while trapped inside this protective shell. Once hatched,
the blastocyst reaches the lining of the uterus, known as the endometrium. Here, implantation begins around the 6th or 7th day.
During implantation, the blastocyst gradually embeds itself into the nutrient-rich uterine lining, establishing the earliest physical connection between the mother and the developing embryo.
Successful implantation is one of the most critical events required for a healthy pregnancy. During implantation, the trophoblast differentiates into two specialized layers. The cytotrophoblast consists of actively dividing cells that continually produce new trophoblastic cells. Surrounding it is the syncytiotrophoblast, a multinucleated invasive layer that penetrates deep into the uterine tissue and establishes contact with maternal blood vessels.
This layer begins producing human chorionic gonadotropin, HCG, the hormone detected by pregnancy tests.
HCG plays a vital role in maintaining early pregnancy by supporting the corpus luteum, which continues producing progesterone until the placenta becomes fully functional.
At the same time, the inner cell mass differentiates into two distinct layers, forming the bilaminar embryonic disc.
The upper layer, known as the epiblast, gives rise to the entire embryo, while the lower layer, called the hypoblast, contributes primarily to the formation of the yolk sac.
During this stage, the amniotic cavity develops above the epiblast, and the primary yolk sac forms beneath the hypoblast.
The amniotic cavity will eventually fill with amniotic fluid, providing mechanical protection, temperature regulation, and freedom of movement for the growing embryo throughout pregnancy.
Major developmental event is gastrulation, which begins during the 3rd week of embryonic development.
This remarkable process transforms the simple bilaminar disc into a trilaminar embryo through the formation of the primitive streak.
Cells from the epiblast migrate inward through this streak and reorganize themselves into 3 retition of every tissue and organ in the human body.
The ectoderm develops into the nervous system, brain, spinal cord, skin, hair, and the sensory organs. The mesoderm forms the heart, blood vessels, blood cells,
muscles, bones, connective tissues, kidneys, and reproductive organs. The endoderm gives rise to the lining of the digestive tract, lungs, thyroid, and many other internal organs.
Every complex structure of the human body can ultimately be traced back to these 3 embryonic germ layers. Soon afterward, a highly specialized process called neurulation begins. Part of the ectoderm folds inward to form the neural tube, the embryonic structure that later develops into the brain and spinal cord. Proper closure of the neural tube is essential for normal neurological development. Failure of this process can result in severe congenital disorders such as spina bifida and anencephaly.
This is why adequate folic acid intake before conception and during early pregnancy is strongly recommended by healthcare professionals worldwide.
Following neurulation, the embryo enters the stage of organogenesis, during which all major organs begin to develop. By the 4th week, the primitive heart starts beating and circulating blood.
The early brain rapidly expands into distinct regions, tiny limb buds appear, and the foundations of the eyes, ears, lungs, liver, kidneys, and digestive system begin to form.
For the following weeks and months, these organs continue growing, differentiating, and maturing until they are capable of supporting life after birth.
This extraordinary sequence of events demonstrates the remarkable precision of human embryology. From a single fertilized cell, countless rounds of cell division, differentiation, migration, and molecular signaling ultimately create a fully developed human being. It is one of the most complex and awe-inspiring biological processes in nature, revealing how life emerges through an extraordinary combination of genetic,
cellular communication, and developmental biology.